Well, good morning, everyone, and welcome into another very exciting Exploring by the Seat of Your Pants broadcast. If you are joining us for the first time today, we are all about bringing conservation, adventure, and science into classrooms around the world, and everything we do goes to our YouTube channel. So if you want to check out like 4,000 broadcasts with the coolest scientists and explorers on planet Earth, I can't encourage you enough to go check out that. It's like a living library of the best and brightest on planet Earth. And I'm really excited of all days you're joining us today because we are back in touch with our amazing Gairdner series. So the Gairdner Foundation showcases and celebrates the very best in biomedical and health sciences. Many of their laureates have gone on to win the Nobel Prize, so they really are some of the most spectacular people on planet Earth. And I'm really thrilled, even within that series, to welcome back Dr. Rodolphe Barrangou. Now, he is the most elegant describer of CRISPR, one of the most revolutionary technology sciences in the last few decades on planet Earth. There's a reason we've had him on 4 times now, and so you are in for an absolute treat as we do a deep dive into CRISPR, what it's all about, how it can impact our future, particularly within the food sciences realm, which Dr. Rodolphe is particularly keen on. And so without further ado, I'm going to zip it. I'm going to turn you over to him. Thank you so much for joining us again, and welcome back to the broadcast, man.
Well, Jesse, thank you for having me. Hi, everyone. It's such an exciting time for science in general and CRISPR in particular. And in many ways, 10 years in, we're just at the end of the beginning. And as a matter of fact, every week there's new CRISPR news coming out. And just this past week, for the first time ever in the CRISPR world, people were able to do an N-of-1 study and use CRISPR genome editing technology to cure a single infant with an orphan disease for which there's no standard of care. So we're in it for a treat here. And to your point, CRISPR is exciting, genome editing is exciting. There's many applications and many implications. And I'm likewise thankful to the Gairdner Foundation to give us the opportunity to celebrate great science, to convene together, and then hopefully I'll help inspire the next generation of great scientists that we have here. On background here for some of the people online, to give you some context, I actually have had 5 different careers so far in my life. I've had one career as a student. I went to school a lot for a lot of years to get a lot of degrees, and learning is great. And what's good about school that sometimes people don't appreciate is you learn skills that you will use for a lifetime. And I would not be the scientist that I am and have had the success that I have had if not for all the schooling that I did. Second career I spent the bulk of my early career in industries. I actually worked in the real world for more than 10 years before I went to academia. And I had the great privilege to bring CRISPR to practice in the real context and understand what I call translational science, which is the challenge to bring great science and great technology to the real world and get products. And I'm going to tell you right now, as you'll hear in the next 30 minutes, I can guarantee you have consumed individually a CRISPR-enhanced product already in your life that I launched when I was in industry, probably unbeknownst to you. The third career I've had, which is one of the major hats I'm wearing today, is as an academic running a lab as a professor at an institution called NC State in the US on the East Coast in beautiful North Carolina. And in the past 12 years, I've had the great privilege to run that lab. And in the past 10 years, I've had the great privilege to partake in some of the Gairdner Foundation outreach programs. And it's so fun to not just do the science in the lab, but also go into the classroom in person or online like today and bring science to the next generation of scientists and train the next generation of scientists. Now, thankfully, because of CRISPR, you hear about that a little bit too, I'm also a serial entrepreneur. I've had the great privilege to build 5 different CRISPR companies in the past decade. And I'm very passionate about not just doing the science, but bringing that technology to the real world. And thankfully, some of those companies have dosed patients. Some of those companies have planted CRISPR-edited plants. Some of those companies are going to dose more patients next year. And it's great to see the speed and space and scale at which we can take great science and great technology to the real world. And then, of course, because CRISPR is so exciting, I also have a whole career as an investor, not just to invest in people, but invest in great companies and great technology that are changing the world. So on that note, with that background being told now, I'm here today to tell you about what I call the innovator's dilemma. I'm a scientist, I'm an engineer, I'm an entrepreneur, I'm an academic, and many more, and much more. And the challenge of innovation is to bring great science and turn it into great products. And it's one of the hardest things on planet Earth because we have to have great science to start with. And sometimes we can take some of that great science into very useful technology. So CRISPR science to CRISPR technology, and then apply that technology to do genome editing in organisms of interest and eventually productize it and commercialize it. And it's not just enough to have a good product. That product needs to be accepted by inventors and regulators and the public and consumers and farmers. And one of the key lessons for today is no matter how good you are, how smart you are, how brilliant and innovative you are, we all have to work together as communities to transcend all those steps and achieve everything that we have to do to turn great science into great products. And whether you're in the academic world in the future, your industrial world in the future, in the government, nonprofit world in the future, or you're just, part of society as a consumer, we all have to do our part to enable great science and great technology to solve the great problems that we have on planet Earth. So let's talk about CRISPR for a minute. Before I tell you what it is, I'm going to tell you what we are. And I mentioned already that we are at the end of the beginning. And what's interesting is 10 years ago, CRISPR pretty much did not exist. And in the past decade, we've seen a remarkable explosion of that disruptive technology, and in some ways, democratization of that technology. On background, there's about 250,000 labs in the world that use CRISPR. And CRISPR, the technology, has been deployed to over 100 countries. So it's very competitive. There's over 50,000 people that have already written a CRISPR paper. And you'll note Canada, is near the top here. It's very international. Most continents, have countries and people and labs that do CRISPR. It's very biased. About 80% of all that work is focused on human medicines and maybe about 5% in ag. And there's all kinds of things we can do with CRISPR. So the point is, it's a very powerful disruptive technology. Now, how do we get here? So it starts with the science. As a matter of fact, the reason I'm here today and the reason I was recognized by the Gardner Foundation is because of the early work that I did on basic CRISPR science to understand what CRISPR is in nature and how CRISPR works. And I'm not going to give a lecture on CRISPR science whatsoever. All you need to know is a very mysterious adaptive immune system in bacteria that allows bacteria to fend off viruses. And the way it works is by enabling bacteria to have DNA-encoded, RNA-mediated nucleic acid targeting. Long story short, CRISPR is a pair of scissors or molecular scalpel that allows bacteria to cut viral DNA when they get infected. That's all you need to know about CRISPR. Now, what's remarkable about CRISPR is not just the great science of what it is and where it comes from, is how it works, but more our ability to use CRISPR as genome editing technology. And the reason why Jennifer Doudna won the Gardner Award in 2016, the reason why she won the Nobel Prize in 2020, to Jesse's point, is because she invented a CRISPR-derived method to have a molecular scalpel that allows us to cut DNA virtually in any organism on planet Earth. And what happens when you cut DNA is the same thing that would happen if you use a molecular scalpel and cut yourself on your skin. There's a little scar that occurs at the site of cleavage. And in DNA, that little scar is a change precisely at the site of cleavage. And what you can do is use CRISPR as a control F function in text. You can imagine, maybe you've read Sir William Shakespeare prose in your English literature studies. You can look for a sequence in a large genome, control F, cut precisely where you want to make the change. And then once you have that cut, you precisely edit the DNA at the site of cleavage. Imagine control F to be or not to be, that is the question. You can control CRISPR to cut between that and is, to be or not to be, that is the question, and you can change the text right there. That's the question, hashtag the question. You can add a word, remove a word, or change a letter. Now, in the past decade, those 50,000 people that have written 40,000 papers on CRISPR have developed a whole CRISPR molecular toolbox, and we're going to talk to you about, that allows us not just to change the text, but also to change the punctuation. So we can now change DNA as a sequence. We can now turn on DNA. We can turn off DNA. We can turn up DNA expression. We can turn down DNA expression. And we can not just change the genome and the transcriptome, but also the epigenome, the epigenetic state, which is how the chemistry of the DNA is changed based on environmental cues. The point I'm making here is we cannot just say to be or not to be, hashtag the question. We can turn things on or up or on or off. To be or not to be, the end. to be or not to be? That's the question, right? We really have an amazing ability now to use that molecular toolbox to change DNA virtually any way we want in any organism we want. And in the past decade, I and my 50,000 colleagues that do that all day, every day, have had great success at rewriting the code of life in organisms across the tree of life, whether you work on viruses or bacteria or yeast or model organisms we study in a lab setting. Anything you can think of has had its DNA changed using CRISPR. Jesse mentioned food and ag, and I'm going to talk about food and ag in a few minutes here, but any crop, any plant, any vegetable, any fruit, any livestock, any kind of meat that you like to consume, real or pretend meat, has had its DNA edited by CRISPR to change the functionalities, the flavor, and or the texture of the food. And then, of course, the top of the pyramid, predictably and as expected and hoped, CRISPR has made miracles in the clinic by curing diseases and dosing patients. I'll talk about that in just a few minutes. Now, we're looking at butterflies here. And I trust that most people on planet Earth, regardless of whether you're an animal lover or not, whether you like to look at them or eat them or pet them or play with them or observe them, I think butterflies are usually pretty popular. And what's remarkable about butterflies is that the wing pattern of a butterfly is the equivalent of the human eye iris. It's unique in the world. No two butterflies in the world have the same wing pattern. To illustrate the power of CRISPR, you can take some cells out of this butterfly and deliver in it a CRISPR that would control F and edit a gene called yellow. And predictably, yellow is the gene that's responsible for the yellow pigment in the wing pattern of butterflies. So you can take a cell out of the butterfly, control F yellow, turn off yellow, and grow a smaller butterfly like this that remarkably has the same exact DNA, except for one change in the yellow gene, and remarkably and predictably has the same exact wing pattern as the other butterfly, except without the color yellow. You can do that with the color black. You can do that with any color you want. The point I'm trying to make here is that if you can use CRISPR to recolor the wing pattern of butterflies, I would make the argument that there's no limit to what we can do with CRISPR, but our imagination. And thankfully, the world is pretty imaginative. And us scientists collectively have illustrated in the past 10 years how we can deploy CRISPR to change the eye color of insects, to change the color of fish, to humanize pigs and make them resistant to disease, to grow trees that are more sustainable, to change fat, protein, or lipid composition in livestock, to make cats hypoallergenic, to make crops more sustainable, to edit the composition of lamb, to change just the one eye color in one animal, and then, of course, to dose patients and cure human disease. If there's a species of interest to you, if there's an animal, a plant, an organism you can think of right now, I can pretty much guarantee you that people like me have used and deployed CRISPR to change their DNA. And CRISPR is about 12 years old right now. So some of you might be around 12 years old, maybe a little bit older, maybe a little bit younger, depending on who's online here. And you can think of CRISPR as an amazing 12-year-old prodigy. It's maybe the most prodigious, disruptive technology we've seen in molecular biology ever. Some people would argue cloning, some people may argue PCR, but I would argue our ability to change DNA, rewrite DNA, recode DNA, and change the code of life. It's very powerful. And the reason this 10, 12-year-old technology is so prodigious is because it's programmable, specific, transferable, efficient, precise, affordable, quick, multiplexable, and scalable. It's just amazing. But it's only 12 years old, so we need to thin out a little bit, get a little bit more reliable, throw fewer tantrums here and there, and be more predictable. But for all intents and purposes, as a 12-year-old technology, is already FDA approved to cure disease. So the technology may not be perfect. We're still working on it, making it better, making it leaner, making it cheaper, making it more affordable, making it more scalable, making it more precise, making it more efficient. But it's still good enough to be a world-class Olympian, gold champion. Now, there's a lot of people like me and others working on making CRISPR better with AI, making CRISPR better with synthetic biology, making CRISPR better by mining diversity in planet Earth, using structure-informed mutation, mutagenesis, and on and on and on. And of course, CRISPR does the cleavage, but we also need to understand how the DNA repair mechanisms occur so we can not just predict the cleavage, but also predict the outcome. And then on a practical basis, for those of you who worried about medicine and deployment in patients, it's not trivial whatsoever to deliver CRISPR to all tissues. So it's easy to deliver to blood, it's easy to deliver to muscle, it's easy to deliver to the eye, it's easy to deliver to the liver, obviously, but there's still parts and tissues of humans that are hard to deliver to, like the brain, and other tissues that are more finicky. That being said, though, those 50,000 people like me who've published 40,000 papers like me have been very hard at work in the past decade to apply and deploy CRISPR technology for all kinds of practical applications and commercial pursuits. As a matter of fact, there's a whole business of enabling people who do science. You can buy CRISPR tools, CRISPR toys, CRISPR enzyme, CRISPR plasmids, CRISPR kits, CRISPR, everything that you need to do this in the lab. I'm not going to talk much about that today. What's interesting to me is that any bacterium, any yeast, any algae, any organism used in biotech industry to make food, to make bioproducts, to make bioenergy, biofuel, or household care products have had their genomes enhanced using CRISPR. So for example, laundry detergent is one of the most used things across planet Earth. The difference between the cheap laundry detergent and the expensive laundry detergents is enzymes. Right? The cheap laundry detergents have great scents and great flavors and great chemicals. The expensive laundry detergents also have enzymes that target specific stains. So if you're an athlete and you have a green stain on your shirt or your socks or your shorts, because there's cellulase, there's grass-derived stains there, you can actually engineer cellulases to very specifically cut out that stain. Not make it go away, not make it faint, but make it completely disappear. Those enzymes are made using CRISPR. So next time you go do your laundry, think of CRISPR. Of course, in agriculture, any plants for crops, any animal for livestock, any microfermentation, any tree for forestry, flowers, ornamental, grasses, aquaculture, seafood, fish, check, 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 check. All those organisms have been enhanced and bred using CRISPR. And of course, in therapeutics, the possibilities are left to our imagination: gene and cell therapies, antimicrobials, microbiome engineering, cell and immunotherapies for cancer, engineering human tissues to do cross-species transplantation from livestock like a pig into a human, think kidney transplants and the like, gene drive diagnostics. We can do that for pet care and animal as well. The possibilities are limitless. So let's talk about some products. The first product ever commercialized using CRISPR technology is actually yogurt and cheese. Since 2011, about 14 years ago, we launched the first ever CRISPR-enhanced dairy starter culture that is CRISPR-enhanced for phage resistance to make yogurt fermentation more sustainable, to make cheese fermentation more sustainable. That means that if you've had a bite of yogurt, a bite of cheese, a slice of pizza, a tray of nachos, a cheeseburger, poutine, anywhere in the world since 2011, I can guarantee you have consumed a dairy product that was fermented using a CRISPR-enhanced starter culture, and you're welcome for that. But the same technology has been used using an engineering approach to change the composition of fruits. I think like non-browning mushrooms, non-browning fruits, non-browning kale, and the like. The FDA just approved last month a CRISPR-edited pig for viral resistance. Think of hornless cattle. Think of more sustainable crops. Think of higher yield corn, soy, rice, potatoes, tomatoes. Again, I mentioned fish, I mentioned cheese, I mentioned cattle, fruits and vegetables, using CRISPR to enhance the nutritional value of crops, right? And if you think about our need to build a more resilient, sustainable, and healthier food supply chain, you can imagine how great science and great technology is the very foundation for that. Now, if you really want to understand where we are right now, in the last 5 years, since 2020, or maybe 2019, there was a pre-CRISPR element there. But in the last 5 years, remarkably, corn and soy and tomatoes and fish and cattle and lettuce and mustard greens and on and on and on have been developed by a slate of different companies in different regions, targeting different diseases, different functions, different phenotypes at different times, at different spaces, and different markets to bring ag products to reality. And as a matter of fact, the prediction right now is that over 100 million farms will plant CRISPR edited material by the end of the decade. And I want you to think about that number just for a minute, right? 100 million farms are predicted to use CRISPR seeds and plant them to grow human food by the end of the decade. Now, I'm very excited about the deployment and the potential of CRISPR for genome editing in the clinic. And what's important here, and I wrote a whole essay about this a while ago, is CRISPR doesn't just rewrite DNA, it's rewriting in real time the future of medicine. Right? George Daly, who's the dean at the Harvard Medical School, is compelling us to welcome a new era, the era of gene editing in translational medicine. For those of you who know someone in your network, in your family, who may have a genetic disease, maybe something like sickle cell or beta thalassemia, note that the first ever CRISPR-approved drug, Casgevy, which is now FDA approved, right? Like in available select clinics, you can, with a single dose, take cells outside of a patient, edit their DNA to correct the genetic disease, expand the cell population, infuse those cells back into the patient, and cure the disease in a single dose. It's remarkable, right? Now, you can't just take cells out and edit them ex vivo and put them back in the patient. You can also deliver CRISPR directly to tissues of interest. I mentioned delivery to the liver, and one of my companies has shown that we can use CRISPR-Cas9 in vivo in patients and deliver CRISPR to their liver to knock out genes that are responsible for TTR and amyloidosis. And then again, just last week, right, Kira Musunuru showed that an N of 1 patient who was 6 months old and had a rare, almost unheard of genetic disease, within months, you can work very hard with a lot of people, a lot of resources to develop a new drug that doesn't exist and correct a single genetic disease in a single patient. And that's opening up a whole new world, according to Peter Marks from the FDA as of last week, to show that we can now do N of 1 therapy. Think of personalized medicine at its best. We can go into one patient with their unique genetic content and customize and personalize a cure that will save their life. And that's opening up the whole world to extend gene medicines to all patients in need, regardless of your background, regardless of your disease, regardless of your challenges, regardless of your ethnicity, regardless of your DNA sequence, regardless of the nation in which you are, and on and on and on. As a matter of fact, there's over 100 different CRISPR clinical trials underway. Again, in some cases, you go ex vivo, you take cells out of patients, you engineer, correct, edit them in the lab, amplify them, and infuse them back into the blood. Or we can deliver directly CRISPR into patients in the liver, in the blood, in the eye, in the urethra, in the muscle. This particular clinical trial is not as advanced yet, but it's changing now, and on and on and on to do that. There's all kinds of targets that are in play. Of course, cancer, of course, genetic disease, of course, liver disease, of course, heart disease, of course, infectious disease, of course, UTI and other infections. There's all kinds of CRISPR tools we can use for that. I mentioned the scalpel, but some target DNA, some target RNA, some target both. Some do large deletions, some do specific mutations, and then some you can deliver with LNPs or RNPs or viruses and things like this. I'm not going to do that too much. The whole point I'm trying to make here is there's all kinds of companies targeting all kinds of diseases with all kinds of CRISPR tools to target all kinds of patients. And we're ushering in a whole new era of medicine with amazing CRISPR technology. Now, with that power, that enthusiasm, that disruptive innovation, those inventors, those trailblazers, those pioneers, those selfless scientists who are trying to solve all the world's problems have great power under their control as well. And it's important for us to contextualize that work with society. And there's ethical implications that we need to be mindful of. Are we going too slow or too fast? If you talk to ethicists that are very philosophical versus patients that are dying, you get a different answer. And you may have your own opinions as to whether we're going too slow or too fast. Is that good or bad? Is that risky? Is that equitable? Is that affordable? Is that scalable? What are the risk-benefit ratios? Should we prioritize curing disease, or should we prioritize feeding the world? Should we prioritize global sustainability and climate change? Like, different people have different opinions, and I think it's a very healthy debate to have. And we should have the debate now, because now is the time. I think also as scientists, we need to understand how to engage with the public, right? So do we talk to non-scientists too much or too little, depending on where you are and who you spend your time with? Are we talking about genetically modified organism? Is it ethical to change and correct human disease? Are we restoring humanity or changing humanity? Can we trust the people who do that or not? Are they transparent or not? Are they good stewards of the technology or not? And do you want to hear from the scientists? Do you want to hear from the patients? Do you want to hear from their parents or their caregivers? Do you want to hear from farmers or foresters or regulators or governmental agencies or pundits? Right? Are you going to get your scientific news in the scientific literature? Or are you going to get your scientific news on Twitter? You're going to get scientific news from the TV. You're going to get scientific news from scientific experts. And I think we have to really realize how, at a time of communication changes and platform evolution, we need to recontextualize how we talk about science in different realms. And then, of course, we have to educate regulators. How are we going to develop regulations that are adaptive enough and wise enough to define how we're going to regulate or deregulate the use of genome editing in medicine, or the use of genome editing in ag, or the use of genome editing in forestry, the use of genome editing in humans or the environment, in trees versus insects? How safe is it or not? How efficacious and desirable is it or not? And we have to do that on a local basis. We have to do that on a domestic basis. We have to do that on a nationalistic basis. We have to do that on a global basis. And I think you all understand now, regardless of your age and interest, that the world is very interconnected. There's a whole bioeconomy of CRISPR and genome editing that we need to account for. Are we competing with Europe? Are we competing with China? Are we competing with our neighbors next door? And how geopolitically loaded some of those regulatory constraints and commercial constraints and intellectual property constraints and commercialization constraints and investments may or may not be. So as scientists, as consumers, as patients, as inventors, as investors, and as civilians, we have to decide, are we going too fast or too slow? Are we talking enough about CRISPR or not? And how are we going to regulate that moving forward? And that's kind of my call to action for you all today, is whether you want to be a scientist or not, you should have an opinion. Whether you want to do research or not, you should have an opinion. Whether you want to work in industry or not, you should have an opinion. Whether you work in government or not, you have an opinion, because you're in society and you're going to be impacted by those technologies. If you think about your career moving forward, you want to be a researcher and train diversity, be a thought leader and developer and inventor and scientist. You want to work in government to help regulate this or patent this or dictate this or contextualize this or constrain this or invest in this. You're going to be in the real world industry as an entrepreneur, investor, partner, to opportunistically commercialize that and develop that and apply that and translationally translate that into products. Or as a member of the public, as a consumer, as a patient, as a farmer, as a parent, as an opinion provider, do you have an ethical stance on where and how and when and what we should do and how we should do it, and who should do it, and why should they do it, And when should they do it? And where should they do it? And can they be trusted and or untrusted with that technology or not? And you will help shape the next decade of CRISPR and genome editing. And in closing here, again, I want to be thankful to the Canada Gardner Awards and the Gardner Foundation, celebrating excellent science, convening some of the brightest minds in the world to bring science to diverse audiences and inspire the next generation of scientists. And I'm going to urge those of you in the audience here who are curious about science, who have your interest piqued about science and technology. There's so many careers you can have as a scientist, right? You can be like a researcher and do fundamental research in the lab. You can be a teacher or a professor. You can be a speaker or an ambassador. You could be an entrepreneur, an innovative disruptor. You can be a learner and learn about science and learn about the world. You can coach other people to do great science. You can write about science, not just science fiction, but real science. You can write grants and write books and write papers and write proposals and write dissertations and write essays and write opinions. And of course, we need to manage science. We need smart people to help manage all the contextual dimensions that I just brought up. So we need all kinds of people to not just do science, but lead science to the next level. And I would encourage you to look at some of my fellow Gardner laureates, right? Some won the Nobel Prize, some are very Nobel worthy, some are very famous, like Tony Fauci and whatnot. So there's a lot of great people who do a lot of great science all day, every day, who should look up to or look into, and then decide whether they should be a good role model or not, whether they have features, background, interest that inspire you to do science. And also remember that science is here to solve problems, not create them. And I think it's very important to realize that now is a very critical time for science, in a time sometimes of anti-science biases and skepticism. And we have a responsibility as citizens of the world to make sure that we have access to the best science and best technology to solve the grandest problems on planet Earth. So I'm going to thank you for the invitation today. I'm going to thank you for your attention. I'm going to thank you for your curiosity. I'm going to thank you for your forthcoming questions. And I hope that you had the chance to realize there's a lot about CRISPR that is exciting, but there's a lot about science that's exciting way beyond CRISPR. So thank you for having me today, and we'll open the floor for questions.
Thank you so much. I always learn so much, and you've modified the talk every single time you've joined us, and there's always new things to share, which is the essence of CRISPR. It's the essence of science, and so I always really appreciate the chance to talk with you. And again, I really encourage our classes, Gardner.org, at Gardner Awards. You learn from amazing scientists like Dr. Barrangou. There's a whole slew of incredible people there, and we'll be doing a big series with them in October as well, so stay tuned for that in the new school year. We, as always, have a bevy of questions for you. So one, you started off with this first gene-edited infant. And so one of our questions from our high school classes are, how do you get CRISPR into all the cells of like a living person? Like there's a lot of cells. What's the deal?
Yeah, so we can't quite, as of yet, edit all the cells in a whole patient, take a patient and dunk them into a pool of CRISPR and then correct all their cells as of yet, right? I mean, we can't quite do that. So that's why I mentioned the different options of ex vivo and in vivo.
Yep.
So in some cases, what we do is we just take a couple of cells, I mean, a couple of millions of cells, but a couple of cells outside of a patient. Usually we do like a spinal tap, and we take like stem cells that will derive the tissues that we want, and we take them into the lab, and we edit them in the lab outside of the patient, and then we multiply them to some extent, and we reinfuse them into their blood, and they will circulate throughout the body.
Okay.
And they will have enough cell mass to change the disease that you have, right? So that's how kind of like you scale up something without dunking the whole patient in a CRISPR, so to speak. But the other option that we have, which is important,
please,
is sometimes some diseases are tissue specific. So if you have glaucoma in your eye, we just need to deliver CRISPR into your eye and be very local, or you can deliver CRISPR into your muscle if you have a particular muscle disease, or we can deliver CRISPR into the blood if you have a blood disease. And then thankfully I mentioned the liver. The liver is a key organ that detoxifies everything we consume. So everything goes to the liver, and there's a lot of liver specific diseases that have been documented so far. So when we can deliver that to a patient, it goes to the liver and then changes enough liver cells to have what's called a supraclinical effect, where you solve the problem. So that's how you can either ex vivo or in vivo address a larger amount of cells enough to have an effect, or locally have a very specific effect to target a disease.
Very, very cool. And also the very first ever pantomime of an injection in an eye in the history of exploring by the seat of your pants. So thank you for that.
Do not do that at home. Do not.
Yes, no. Let's keep the pens away. Pens away. A question I think we've had in every one of your talks, and I'm sure it's the most common question. I'd be curious if it is the most common question you get, which is, are CRISPR foods safe to eat? Please do tell.
Yeah, I mean, if they were not, you would not be able to buy them and purchase them and get access to them. So, what's interesting about some of those CRISPR foods is we actually use CRISPR to just replicate what happens in nature. We just do it faster, cheaper, and more scalably. So in many ways, most of the applications of CRISPR that I'm mentioning and illustrating today that are on the market right now are indistinguishable from what happens in nature. It's just how we made them, but the outcome is indistinguishable. So that's why they're safe. That's why they're deregulated. That's why they're condoned. That's why they're on the market. And that's why those regulatory agencies have endorsed and condoned the commercial deployment of CRISPR in foods. As a matter of fact, later this year, we anticipate that CRISPR genome editing technology laws that are near the penultimate approval in Europe will be approved by the EU. It's been a long process. It's been a long journey. European has a lot of opinions and difficult ways to regulate what they do and how they do it. But they are kind of like the last bastion in the world, right? New Zealand is also there, almost there. But right now, most parts of the world have CRISPR-approved foods in their ag system, in the ag supply chain. And by the end of the year, the large majority of the world will be on the same boat. What's ironic, just for a minute, is that because the EU has been so slow, as a matter of fact, the scale at which they will allow people predictably to use CRISPR is even bigger than in other parts of the world. So, the more time goes by, it has the advantage of having more science-informed decisions. And it's ironic that the EU, which is right now the conspicuous part of the world that has yet to approve CRISPR, will do so by the end of the year and be the most amenable to doing that. And ironically enough, for those of you who not always think about EU versus Europe, if you go to the UK, there's a bunch of CRISPR food in the UK, right? So UK is in Europe, but not the EU. But because they're not in the EU anymore, with the UK agricultural ministers have done very well. It's actually doubled down on CRISPR, accelerate CRISPR, made decisions to enable the commercialization of the CRISPR tomato that I showed in my talk. And then all kinds of other CRISPRs. There's a bunch of CRISPR companies in the UK that are doing CRISPR in ag because the UK is ahead of Europe, ahead of the EU.
So your way of answering this question leads very nicely into our next one, which is that you're a very enthused guy and you're keen on everything. But is there something you're most excited about? Like, is there that gets you out of bed in the morning?
Absolutely. So what gets me out of bed every morning between 4 and 5 o'clock tirelessly is using CRISPR in forestry to have more sustainable forestry. Because I would argue climate change is one of the biggest challenges that we have right now, one of the hardest problems to solve. And as a matter of fact, about 67 to 70% of all the carbon on planet Earth that's bioavailable is captured in plants called trees. If you haven't taken a tree down, you can appreciate how heavy trees are. And students nowadays don't use books the way we used to when I was young. But if you have a backpack full of books, it feels very, very heavy, right? And all that paper comes from trees. And it kind of reminds you of how heavy, paper can be. So I'm most excited right now. I mean, we're in the clinic and that's great. Dosing patients is remarkable. We're in the ag world, planting CRISPR-edited crops, and that's remarkable. But we are amongst the very few people in the world, maybe the only people in the world, really committed to deploying genome editing in forestry to breed a more sustainable forest and be more efficient and be more resilient and be more fire resistant and blight resistant and disease resistant and drought resistant and heat tolerant. And it's very, very hard to do so. But we have published milestone papers showing how we can use synthetic biology, AI, and CRISPR to breed more sustainable trees. It took us a while to do that, like 5 years to do that. And then last year, we planted the first ever CRISPR edited tree field trial. This year, we're planning one in a bigger scale, and next year, we hope to plant the first one ever in the Amazon. That is so exciting because it takes so long for trees to grow that we may not get to the scale that we need to get to until 2048.
Yeah.
And we're barely going to make 2050 for some of the goals that we have. So I'm excited about that. I'm very enthusiastic about that. I'm psyched about that. This is what gets me up early in the morning, 6 days a week. But we still have a lot of work ahead of us, and it's going to take a while to get there.
So I have a great final question for you, but I'm going to hold that off for like 1 minute. Just a quick follow-up on the tree, on the forest you have planted. What are your early findings, or are we tracking that, or do we have to wait a few more years? What's the deal?
No, we're there. So we're lowering lignin to make pulping and processing more efficient. So if you think of like hardwood, like I'm knocking on wood right now, it's very hard to turn hardwood into like soft paper.
Yep.
So and the culprit for that is a complex fiber called lignin, which is hard. It's hard to delignify wood enzymatically and chemically. So we use a lot of heat. Oh, very well done. A lot of heat, a lot of chemicals, a lot of enzymes, a lot of energy, a lot of time to break hardwood down into soft pulp that makes paper. And we can use CRISPR to change the overall content of lignin. So we can lower lignin by 20% without really impacting the ability of the tree to grow well and stay straight and be robust. And then we'll also change the fiber composition of how the different lignin fibers are linked to each other to make them less recalcitrant to pulping and processing with heat and enzymes and the like. So we make wood more sustainable and paper more green in some ways.
That's very, very cool. As a person who lives in a mill town now, that the whole industry is built on that.
Paper mills coming to a
revolutionary. Okay, you send it to Corner Brook, Newfoundland, anytime, and we'll get it hooked up. Before we wrap up, a question I think is really important to leave with every time for our students joining today that are in middle school, high school. What would you recommend? You gave a whole bunch of advice at the end of the program. If they're in the middle of their studies, is there anything that they should particularly focus on? Do tell.
2 things. You got to take risks in life. Like, don't be scared to fail. Do what you feel strongly about and have fun on the way. And then number 2, don't do it alone. The most fun you're going to have in life is working with other people. The biggest challenge you're going to solve in life are going to require other people. So fail often and get back up and do it again until you succeed. And then make sure that you don't do it alone, that you have a whole team, a whole squad, a whole, clan of people to do it with, because it's a lot more fun to solve those problems when you have some help.
Yep. I love that this is advice that we get from scientists in every possible discipline in the world, is that you get to work with the coolest people from all around the world and solve great problems together and be creative in a group, and that you're going to screw up all the time. Like, I think when I was a student, when a certain little boy, I was like, I don't want to mess this up. I don't want to get the answer wrong. And the best people that we bring on universally are like, I got all the things wrong constantly. And I learned because of that and got better and better and better. And now you end up with Gardner awardees and more. So thank you, as always, for your passion, expertise, and all of it, making this work. I know you filmed a daily dive the other day, so we're going to have a short little featurette with Rodolphe coming up in the new school year, so you can stay tuned for that, students. But we'll say farewell from there, and I just wish you all a wonderful, wonderful rest of your day, and fail often, everybody. Bye for now, guys. Thank you so much, and we are done live. Thank you kindly for joining.